get information from different omics approach, genomics, and put it into mathematical models to try and understand the factors that shape interaction of microbiota with metabolites, be it nutrients or drugs. Find new pathways that are responsible for these changes in metabolites, and go to the point of having causal inference about what is doing what inside the microbiota. So I want to thank her for the seminar today, also for coming for the IGC PhD program course on host microbe interactions, which is also a course for SymbNET PhD students. And also this seminar is also for the SymbNET community too. Thank you very much.
Thanks a lot, Luis, for this very kind introduction, and thanks for invitation to give the IGC and SymbNET seminar. It's really a pleasure for me to be here today and talk about our work on quantifying metabolic interactions between gut microbiota and the host. And I will cover both drug and not drug related projects in my talk today. So just as an introduction in the beginning, we are born practically sterile, and the microbes that we get from interacting with humans, from diet, with the environment, we encounter throughout our life, especially in the beginning of the life, but basically the microbiome can change also later on. And healthy humans harbor hundreds, if not thousands of species, the majority of which reside in the gut. Gut microbes collectively encode 100 times more genes than the human genome, encoding a large metabolic potential to interact with different chemicals and compounds. And sequencing technologies allow us to relate the genetic diversity of microbes with different aspects of human health. For example, digestion, development of immune system, but also processes associated with mental health, metabolic diseases, metabolism of medical drugs, and processes associated with aging. However, these associative studies linking the gut microbiota composition to different host phenotypes usually remain correlative, where we don't know what is the mechanisms of these interactions and which associations are causative and which are happening just by chance. In my research, I'm mainly focusing on the one aspect of host microbiome interactions, specifically through small molecules or metabolites that can be exchanged between organisms, between host and microbiota, or between members of the microbiota. And today, I will talk about two projects in which we aimed to answer the question of how much bacteria can contribute to the host metabolism and how common such microbial contribution to the host metabolism can be. The first project, we started answering this question on specifically for medical drugs. And drugs are not only relevant for human health, but also are a nice model for studying host microbe interactions, because drugs are produced neither by the host nor by the microbes, and then can be introduced into the system in a more controlled way. So we started with one particular drug, Brividine, which is an antiviral compound taken orally. And this compound is known to be metabolized to a metabolite bromovinyl uracil, or BVU. And this metabolic conversion can be performed both by the host and by the microbes. And it was known before, but we also confirmed it experimentally by performing human and mouse liver assays with Brividine, and showing that human and mouse liver can convert drug to drug metabolite. And we also showed it for microbes, where we incubated Brividine with fecal material from either human and mice, and showed that fecal material of human and mouse also can perform drug-to-drug metabolite conversion. And now when we confirmed that microbes in the gut can perform this conversion, we were wondering whether we can identify which microbes specifically can perform this metabolic function. To answer this question, we went to our collection of isolated gut microbes in the lab and incubated individual microbes with Brividine in the lab and tested which of these microbes can perform drug-to-drug metabolite conversion by measuring drug and drug metabolite concentrations over time. And as you can see with these colorful plots, we tested a bunch of different bacteria, and they had different capacities of performing this metabolic conversion, with the fastest metabolizer being Bacteroides statiota omicron, showing here in dark green. And this was actually quite a lucky outcome for us, because Bacteroides statiota omicron is a quite common human gut microbe, but moreover, we can also perform genetic manipulation of these microbes. So we could use Bacteroides statiota omicron to try to find the metabolic gene that performs drug-to-drug metabolite conversion. For Bacteroides statiota omicron, we have a transposon library in the lab where we have several thousands of strains with random mutations in different genes in this bacterium, and we incubated this library with the drug and found one clone here in orange that lost the capacity to convert drug to metabolite. And we then perform genetic follow-up to show that this gene is necessary and sufficient to perform the conversion, because the mutant of this gene, BT4554 here in orange, is not able to convert drug to metabolite. So now we had quite a solid experimental evidence that we know which bacterium and with which gene can perform drug metabolism. But the big question remains, does it actually play a role in the organism? And to check whether bacterial drug metabolism plays a role in the animal, we went to the germ-free mouse models, which lack any bacteria in and on their body and can serve as microbiota negative controls. So this photo represents basically the germ-free mouse isolator. So mice live inside these bubbles, and everything that gets into this isolator is sterilized. So we make sure that microbes have absolutely no microbes in there. But we could use this germ-free animals to colonize them either with the mutant or the wild type of Bacteroides statiota omicron lacking the gene of drug metabolism, and see whether the single bacterial gene deletion will affect drug kinetics in the animal. We measured Brividine in different tissues, and here I'm showing you in orange the mouse colonized with the mutant bacteria, and in green mouse colonized with the wild type bacteria. And as you can see here, although in the serum, the drug kinetic profile hardly is different between the two mouse groups, we see large differences in the large intestine, where no drug is detected in the wild type colonized mice, and a lot of drug is detected in the mutant colonized mice. Moreover, for the drug metabolite, which is conditionally toxic, we found that both in the large intestine and in the serum, we find significant differences in drug metabolite concentrations between wild type and mutant mice, with wild type mice having much more toxic drug metabolite both in their guts and in the serum. So to quantify specifically microbial contribution to drug metabolism, we developed a pharmacokinetic modeling approach where we consider different processes of drug metabolism in the body. So when drug enters the body, it can be either absorbed from systemic circulation, from small intestine into systemic circulation, where it can be metabolized by the host, or it propagates down the gastrointestinal tract and gets eliminated from the system. And we can describe these processes with ordinary differential equations, example of which I'm showing you here on the top, where, for example, for drug concentration in serum, we define it as being proportional to drug absorption, drug metabolism by the host, and drug elimination. And for the we can parameterize this series of ordinary differential equations for the host parameters using the data from mutant colonized mice, because in mutant colonized mice there is no microbial contribution to drug metabolism. And then for the mice that are colonized with wild-type bacteria, the drug that remained in the large intestine can be metabolized by bacteria. And this bacterial drug metabolite can be either propagating down the gastrointestinal tract, or it can be converted, it can be absorbed into systemic circulation. So, as I said, we fitted the host parameters with the data from mutant colonized mice, and we used the data from wild-type colonized mice alone to parameterize the microbial parts of this drug metabolism. And this model allowed us to predict how much of bacterial produced drug metabolite will be absorbed into the systemic circulation. And together with our estimates of how much of drug metabolite produced by the host, shown here in black, we can predict what will be the difference between wild-type and mutant colonized mice. And these differences match experimental data very well. So indeed, the mutant colonized mice in orange and the wild-type colonized mice in gray, excuse me, have different drug metabolite kinetics, and the model allows us to estimate that bacteria contribute 70% of the toxic drug metabolite exposure in the host. And yes, of course.
So while these are lovely experiments, losing the rest of the microbiome seems to be losing a very large number of potentially modifying factors. Is it fair to generalize from this very highly simplified experimental model to what happens when you have 300 other species of bacteria present?
I think it depends on the drug. So in this case, of course, it's a very clean system where we have one bacterium, and we could show that this single bacterial gene affects drug and drug metabolite kinetics. But this approach would also work for a native microbiome. And in this case, we don't know which bacteria, we don't necessarily know which bacteria are performing the function, but we can model it as microbiome as a whole and show whether microbiome as a community can perform drug metabolism and whether this would affect drug kinetics in the body. And this is actually what I'm showing you on the next slide. So we wanted to see how generalizable this approach is. So we decided to apply it to a different drug, which has also more complex kinetics than Brivudin, that was metabolized by only one metabolite. So we also performed the same approach to study gut microbiota contribution to metabolism of clonazepam, which is an antipsychotic drug. And this drug can be metabolized to 2 different metabolites by the host, actually to 3 different metabolites in 2-step reactions, and it could also be glucuronidated and deglucuronidated. And the reactions highlighted here in green are also performed by the microbes, but in this case, we don't know by which microbes, but we know they're performed by the entire microbiota. So I'm not showing you the details of their experiments, but the principle is the same as for single microbes. Here we compare the Jomfrey mice to a mouse colonized with the native microbiota, and we show that for clonazepam and its metabolites, we also see differences in clonazepam metabolite kinetics in the serum. And in this case, microbiota also contributed about between 60 and 70% of metabolite exposure in the serum. So with this model at hand, we can also try to identify factors that affect microbial contribution to the host metabolism. Here, for example, we can fix the host drug metabolism and simulate in silico what will happen if we have different bacterial drug metabolism parameters that would correspond to microbiota with different metabolic capacities, and predict how much of drug metabolite will be observed in serum. We can do it for several parameters simultaneously. For example, this 3D plot shows how much brividine exposure will be observed at different values of bacterial and host metabolism parameters, and the color shows how much of this metabolite in serum will be contributed by the bacteria. And we could also use this model to explain some of the findings that were initially puzzling for us. For example, as I mentioned before, for brividine drug kinetics, we saw literally no difference between a drug profile between wild type and mutant colonized mice, but large differences in the drug metabolite levels in the serum. And we used we simulated how parameters of our model affect drug and drug metabolite levels in the host, and we showed that basically the drug kinetics in our model is influenced mainly by the host parameters of drug metabolism, whereas drug metabolite levels are influenced by both host and bacterial parameters. So here different parameters are on the left, and the wider the bar, the more the bigger the difference in the metabolite or drug concentration will be observed as you vary the parameter values. Yeah, there is a question.
So in your mutant colonized mice, you see an increase of BVU very much at the beginning that goes to the same height as the wild type mice, but then the persistence of the BVU is longer. So you get metabolism of the drug, but then it persists for longer. So what is the explanation for that?
So you're asking why in the BVU, in the BVU kinetics, there is a longer exposure, like there is a longer, basically, distribution of this metabolite.
Yeah, so the concentration goes up, so it's metabolized the same way in the beginning, in the first hour or so.
Yeah.
And then it stays for longer. It's not.
So what, oh, I'm sorry, that's not what I wanted to do.
Something.
So I think that the answer is that the BVU concentration is affected by two main factors, the host metabolism and the bacterial metabolism. And the host metabolism happens much faster because the brividine gets absorbed from the small intestine within the first half an hour or so. There's a high peak, as you can see here. There is a high peak in brividine, and basically the BVU concentration that we see in the first hour is the one that is coming from the host. And then the brividine that gets propagated to gastrointestinal tract and gets metabolized by the microbes, it's delayed in time because the drug needs the time to get down and get metabolized. So basically this peak that we're seeing here at 3 hours is the microbial contribution on top, which is delayed compared to the host contribution.
Thank you.
Oh, sorry. Yeah, I think I'm a bit lost on the modeling approach. So I think you showed some kind of mass conservation equation, right? And there's some parameters there, right? So are these all the parameters in your model? Maybe that's.
So these are pretty much all the parameters. I mean, it's this is pretty much all the parameters in the model, and that basically describes drug and metabolite concentrations in each tissue. So we have 6 sections in the large intestine that we model, and 1 section which is kind of systemic circulation, which is like everything that happens outside of the gut. So we merge basically all the host processes are happening in this 1 compartment.
And how many variables? So you have so these are the parameters, and how many equations and variables are we talking about? That you're roughly.
So it's roughly about 8 equations for the drug, and also about about like 4 for the metabolite. So it's around 12, 15 equations that we're using to describe these processes. And I mean, it's not much different from the classical pharmacokinetic models that are used to describe drug metabolism in the human or in animal models in, for example, pharmaceutical industry. But the main difference here was that we could have the experimental way to separate host and microbe contribution. So we had this host with microbe that has no microbial contribution and host with microbe that had microbial contribution. So we added, so this allowed us to add microbial metabolism in this model, and also provided with the data that was necessary to estimate the parameters of microbial contribution in the gut. And the combination of experimental data that we had with a model that includes explicitly gut microbiome contribution was basically what allowed us to estimate.
And it allows you to estimate these parameters, and then you can make predictions with them. Okay, thank you very much.
Okay, so basically this simulations allowed us to show that in this model that parameterized by for brivudin specifically, regardless of bacterial metabolism coefficient, here very low in blue or very high in yellow, we will see large differences in both drug and drug metabolite concentrations in the gut. You see here flat line for green and for blue, and a lot of brivudin produced by the orange, but there will be no differences in the serum for the drug, whereas there will be still large differences for serum for the metabolite. And basically, we tested which of the parameters in our model are the bottleneck of microbes affecting also the drug levels, not only for drug metabolized levels. And not surprisingly, the bottleneck is here in this tornado plot, which is drug absorption from the large intestine, because small intestine and large intestine have different properties, and different molecules can be prone to be absorbed in different tissues. So in this case, drug itself was not absorbed from the large intestine, but modified drug metabolite was actually absorbed from the large intestine, and that's why we saw this differences in BVU profile with the delayed concentration of BVU in the mice colonized with microbiota that was performing the metabolic function. So with this, I want to summarize the part on the drug metabolism, where our experimental setup allowed us to experimentally separate host and microbial metabolism and develop a mouse model that provides great data set to quantify host and bacterial contribution to a shared metabolic process in vivo. And with this model, we could also investigate factors affecting microbiota host metabolic relationships, and make some predictions on what properties of the drug or host and bacterial metabolic capacities would affect the drug kinetics in the mouse body.
Yeah.
Yes.
Now, I guess that you are changing gears. So you are using drugs that are used, I guess, for therapeutic effects, right? So can you predict, because of the microbiome, if it's going to be effective, or if the or if the drug is going to have side effects or something like that from the microbiome?
So in principle, yes. So what we learned from here is that, it depends on the drug, right? Because it depends on the properties of the drug, whether drug metabolite is toxic or not, whether drug is administered in an active form or not active form. So in this particular case, microbial contribution was actually contributing to drug toxicity because this BVU metabolite is potentially toxic. But there are other examples where microbial activity actually contributes to drug efficacy because either microbes are activating the drug in the gut, or microbes are there are some drugs that undergo glucuronidation, and then they get secreted back into gastrointestinal tract, and microbes are deglucuronidating those compounds, and the drug can get back into systemic circulation. So from our data alone, I would say we cannot really predict that. We can use the model to basically simulate what are the expected profiles of drug and drug metabolite, but the actual parameters would be drug specific. So we need other experimental data to test how fast what is the metabolic capacity of microbes to metabolize this drug, what is the drug absorption, and we need to know what is the properties of the drug metabolites.
Yeah, but for instance, can you combine some of these tests with a therapeutic effect, an infection or something like this, and then seeing whether this solves the problem or not?
Yeah, I mean, we could definitely.
I was wondering how we know that the germ-free gut is much more leakier than a regular gut. I was wondering if that affects your model or it's okay because parameters are.
I mean, in principle, it's okay. So for the Brividine example with mutant and wild type, it didn't matter because they both were starting with germ-free, and then we colonized them. So the host was exactly the same. So we didn't have to worry about different host parameters. For clonazepam example that I briefly showed, we did compare germ-free mouse to normal SPF mouse, and there the host physiology is also a bit different because the germ-free gut is much leakier. We ignored it, and it worked. Basically, we could assume that the host is the same, and in this case, the assumption holds more or less true. But it might become an issue that basically the host physiology might be affected, and maybe for some other drugs you need to have to take it into account in another way.
And on a second question, in your host parameters, do you take into account the absorption in organs and the excretion thing?
Yes. So not in all tissues, but definitely I think we included absorption from two sections of the small intestine, and then from cecum, which is the first part of the mouse large intestine. And we assumed that there was no absorption in other tissues, which is an assumption, but I mean, every model needs to have some assumptions. So yeah, I mean, that's one of the main, so the main parameters of host metabolism is absorption from different sections of intestine, drug to metabolite conversion by the host, and drug elimination from the serum, which is kind of lumping together all the elimination processes in the host.
Because if a drug needs to act, I don't know, in the brain or in the liver, or then this model.
Yeah, I mean, in this case, we would need to extend the model. So we already did it for clonazepam because clonazepam undergoes intrahepatic circulation. So we had to add the bile component and model the the process of host glucuronidating clonazepam that would go to bile and go back to small intestine. So if you want to explicitly model some processes in liver and brain, you would need to add them. But in principle, that might be possible.
And I have to be mean and ask this question. Did you check male and female because of the hormones inputs on drugs?
We didn't explicitly test. We didn't explicitly test. So here we had 6 animals per group. I believe they were mixed sexes, but we didn't explicitly test for. And I think for those drugs, we have pretty strong evidence that it doesn't matter because, because for most of these measurements, actually each time point is a terminal time point because you cannot really sample multiple times. So the fact that we have profiles that are quite consistent suggests that there are not so many inter-mouse differences. But I'm sure that for some other drugs, the hormonal levels would potentially play a role, and this would need to be tested. So in the future experiments, we're actually trying to have bigger groups to make sure that we have enough male and female mice. Okay, so I think if there are no more questions for now for this part, I will move to the second part of my talk, which is focusing on the question of how common microbial contribution to the host metabolism can be. I mean, in the drug part, we focused on 1 or 2 specific drugs and showed that microbes can indeed contribute to the levels of drug-on-drug metabolites. And here we were wondering whether we could focus on dietary components and try to estimate post-diet and microbial contribution to the metabolite profiles in different host tissues. So here again, we were using the germ-free mouse model to perform a controlled diet experiment where we designed a synthetic community of 14 gut microbiota species representing 4 major phyla in the human gut. We combined this bacteria in vitro and colonized them into germ-free mice. And then we exposed germ-free mice and mice colonized with this defined community to 2 different diets, high carbohydrate diet and high fat diet for 4 weeks. And after 4 weeks of different diet exposure, we collected metabolomics samples along the gastrointestinal tract from 6 sections along the gut. And we collected samples for sequencing from the cecum of colonized mice for metagenomics and metatranscriptomics. We're mainly focusing on metabolomics. We used 6 different untargeted metabolomics methods and we measured about 20,000 ions and covered about 3,500 compounds from KEC and human metabolome database. And this map here is a KEC map of metabolism, and each red dot shows a metabolite that we could annotate and then demonstrating that we had quite a broad coverage of different metabolite classes in our experiment. So the first thing we looked at was what were the spatial profiles of metabolites along mouse tissues, specifically in the gastrointestinal tract. And we normalized metabolite abundance in the along the gastrointestinal tract. Here, the 6 sections that we measured, and we performed k-means clustering of metabolite profiles that we detected. And we found that there were 6 clusters that were emerging. In this case, I'm showing you one of the mouse groups, the high carbohydrate fat germ-free mice. And we found that when we performed this clustering for each of the 4 mouse groups, we found that the same 6 clusters emerge every time. In each case, the peak intensity of metabolites were roughly in one of the 6 sections. So that's how we interpreted these clusters that are metabolites that are more abundant in different sections of the gastrointestinal tract. So then we wanted to know whether there are specific groups of metabolites that are present in each of these clusters. So we performed chemical group enrichment analysis of metabolites falling into each cluster in each of the 4 mouse groups. And we found that the same types of metabolites were belonging to the same type of cluster in the mice, irrespective of their diet or colonization states. So here the light color indicates which types of metabolites are enriched in cluster 1. And as you can see, all 4 mouse groups are clustering together. And for example, in the first cluster with metabolites that were most abundant in duodenum, the first part of the small intestine, we found metabolites of classes of glycerolphosphorserines, amines, amino acids, purine ribonucleotides, some fatty acids, and we hypothesized that these metabolites are potentially coming from the diet because they're most abundant in the upper gastrointestinal tract. Another cluster I want to highlight is cluster 3 with metabolites peaking in ileum, the lower part of small intestine, and we found here some bile acids and glycosides, this would also make sense in terms of the physiology because bile acids are known to be produced by the host to aid digestion and get secreted into small intestine. So that was the way we explained the clustering of the mouse groups. However, although the types of metabolites were the same for different mice in the same type of clusters, there were still differences between mouse groups within each cluster. And here I'm just showing you three examples of metabolites to illustrate this point. So here for leucine, for example, there were no strong differences between the mouse groups, but for this metabolite 5-aminolevullinate, you see their profile between different mice in the small intestine is the same, but in the large intestine, this metabolite is much more abundant in germ-free mouse that are depicted in lighter colors. And for porphobilinogen, for example, although the profiles look the same, their abundances of metabolites in different mouse groups differ quite a bit. So here again, we wanted to come up with some way to quantify this metabolite profiles along the gastrointestinal tract and potentially estimate the relative contribution to host diet and microbes to this metabolite profiles. So we came up with a very coarse-grained intestinal flux model that assumes that metabolites are propagating down the gastrointestinal tract with their constant flow F. And they can be exposed to host metabolism parameters in the small intestine and in the large intestine. And in the large intestine, we assume that apart from host metabolism, those metabolites can also be modified by microbial metabolism. And in this case, our model assumes the metabolic pseudo-steady state, which is a strong assumption, but the one we had to make. Under this assumption, the mass action kinetic equations that we use to describe metabolite abundance in each of the mouse tissues boil down to a system of linear equations because we assume the steady state, so the right part, there is no difference over time, basically. And this model allows us to in a very coarse-grained manner estimate what are the relative contributions of different parameters or factors in our model that contribute to metabolite profile. For example, for leucine, our model estimates that the negative metabolism parameters of the host and the small intestine are the largest contributors to leucine profile in the gastrointestinal tract. For 5-aminolevullinate, this is mixed. There is a contribution of negative host metabolism parameter in the small intestine, but also some slightly positive host metabolism parameter in the large intestine and negative microbial metabolism parameter in the small intestine that basically explains, or this could explain the differences that we see, that in the large intestine, the abundance of this metabolite is much higher in germ-free mice. So the model predicts that microbes are consuming this metabolite from the large intestine. And for porphobilinogen, all estimated coefficients are positive, and the model in this way predicts a synergistic effect of both microbial and host metabolism on this metabolite level, because when mice are colonized, there is a higher level of this metabolite in the intestine compared to germ-free mice, suggesting that microbes can potentially contribute more to production of this metabolite. And we applied this modeling approach to about 2,000 metabolites that we could stably detect along the gastrointestinal tract. This approach allowed us to group metabolites based on their model coefficients and interpret the sources that contribute to metabolite profiles. For example, for about 40% of metabolites, the main model parameters contributing to the profiles were negative host metabolism in the small intestine, which we interpret as diet-associated metabolites because the host is absorbing these metabolites in the small intestine. There were about 30% of metabolites that had positive host metabolism parameters either in large intestine or in small intestine, suggesting that these are host-produced metabolites, and about 15% of metabolites that were potentially contributed directly by the microbes, because microbial metabolism coefficient was the largest for those metabolites. And then the next thing we wanted to do with this data was identify potential microbial substrates and products. So first of all, we defined microbial substrates as metabolites with negative microbial metabolism coefficient and microbial products, metabolites with positive microbial metabolism coefficient. And we looked for potential known metabolic paths between identified substrates and products in the CAC enzymatic network. For example, we found some examples with entire metabolic pathways connecting potential substrate, for example, glutamate, to potential product citrulline. And here in orange, you see enzymes that are present in microbes and can potentially convert potential substrate to potential product. And as we collected metatranscriptomics data from our mouse cecum, we could actually test whether microbial enzymes along these pathways are expressed and whether there is a differential expression between the two different diets. And we found for this particular example that the representatives of bacteroidetes are the ones that express the highest levels of enzymes along these pathways. So potentially they might be the contributing to the differences in metabolite levels that we saw in the large intestine. And we also found examples where potential substrates, potential product can be converted by a single microbial enzyme. Here, the example that I showed you in the modeling part, 5-aminolivulinate can be converted to porphobilinogen by a single enzyme, porphobilinogen synthase. And we found that the porphobilinogen synthase was present in 4 species in our synthetic community here in the middle lane, but only 1 species, Clostridium scindens, expressed this enzyme in a way that was correlating with the levels of the product. So here, what I'm showing you in this heat map, in the top row is the correlation between microbial abundance and porphobilinogen abundance. So the correlation between microbial abundance and metabolite levels. And there were many, there were like half of the species in our community were correlating with the levels of metabolites. However, if we look at their enzymes, as I said, there was only 4 species that possess the enzyme that can make this product, and the expression level allowed us to narrow down this enzyme to one single species whose expression of this enzyme was correlating with the product. And this is we did it for about 400 substrate product pairs for which we could find a single enzyme that was connecting these substrates and products. And we found that if we look at the distribution of correlations between relative species abundance and the product, metabolic product in the gut, this distribution is very broad and centered around 0. However, if we look at the expression of specific enzymes that are identified by this approach and measured with metatranscriptomics, we found much higher correlations between microbial enzyme abundance, microbial enzyme expression, and metabolic product production, suggesting that this data provides us with information that can narrow down which of the species are potentially involved in the metabolism of specific compounds. So to summarize this part, in our germ-free mouse models and in the diet experiment with targeted metabolomics, we could collect a data set and perform unsupervised clustering that revealed tissue metabolite profiles and demonstrated that the tissue distribution of metabolites is independent of the diet and mouse colonization state. However, there are still large differences within each tissue between different mouse groups, and our intestinal flux model allowed us to roughly estimate a diet, host, and microbiome relative contributions to metabolite profiles along the gastrointestinal tract. And combining this information with metagenomics and metatranscriptomics data from the colonized mice allowed us to narrow down which bacteria and potentially which genes are associated with specific metabolite profiles in the large intestine, and can thus contribute to host exposed to different levels of metabolites in the gut. And with this, I want to thank my collaborators on the project, especially Michael, Rebecca, and Natasha, and Andy, who were mainly involved in the drug metabolism project, and my lab and the funding sources. And with this, I thank you for your attention, and I'm happy to take questions if there are more questions coming.
So I have a question then. So this last part, it's very detailed characterization of a mouse with 14 or 18 bacteria species, right? So how do you go from here now to humans with 1,000 different species and variation between individuals?
Yeah. So a fair point. So the thing is, with the synthetic community, we're trying to have a controlled system where we can actually identify microbial, ideally pathways or genes that are contributing mechanistically to levels of certain metabolites in the host. And we believe that if we find the genes, that some of them would be universally applicable also to more complex microbiotas. So maybe the microbes might be different, but gene conservation will be like the genes will be conserved. So if we find this porphobilinogen synthase gene, for example, which we now hypothesize are affecting porphobilinogen levels in the host, we can then go to the complex human community and try to measure whether the abundance of this gene or the expression of this gene is associated with porphobilinogen levels in the human, for example. And if we start with human or with native mouse communities, there's just too many too many features, too many microbes, too many genes to find mechanistic links. So we believe that like in a controlled smaller system where we know which microbes are there and we can really attribute gene expression to each microbe, we can find genes and pathways that are generally that are generally that we can generalize to human populations as well.
Okay, we read it afterwards. We have another one here.
Thanks, nice talk. I have a question that is mostly curiosity. So this gene that you found to be responsible for converting in the first part, is it this gene that is already described, its function? And if it's affects the fitness of the bacteria inside of the gut?
Yes. So this gene, I mean, it was not described before. It was not well described before we found that it's metabolizing the drug. But it was kind of described in the same in almost in the same time. So we know now a little bit more about this gene. And it's not only metabolizing the drugs, it's also metabolized either. So the drugs that we tested, Brividine is a nucleoside analog. So basically this gene is also involved in nucleoside metabolism and can metabolize purine nucleosides. And I'm not sure whether it has a phenotype, but this probably someone has checked it. I just don't remember it has a phenotype in the mouse or not.
I don't know if it's possible to think some kind of strategy to change the composition of the microbiota or replacing this bacteria to have some kind of therapeutics. Or do you think it's possible?
I mean, I think we are still somewhat far from targeted manipulation of microbiota. Like that you really can take out this microbe or all microbes with this gene, because in this case, we had a clean system, but there is also other bacteria that have this gene, so they might be also performing the drug metabolism. So I think that's definitely a goal and kind of an outlook that's having more and more of this information, we will be able to design potentially also ways to modify microbiome. I think at the moment it's more realistic that we can use it as a diagnostic, that you can predict how much of metabolism will be performed by the microbes based on the composition of the stool.
So we have a question online about what is the age of the mice that were tested in these studies.
I think it's between 6 and 12 weeks.
So a question from ignorance. Is there a large body of information about who is metabolizing the therapeutics that we receive?
So it's getting larger, the body of information. I think it was not that, I mean, the case reports that microbes contribute to metabolism of medical drugs have been reported for decades, but like very sporadically.
Just to add, sorry, before you finish, to what extent are beneficial effects of therapeutics derived from bacterial metabolism? I mean, is that knowable now?
Benefit, like where microbes are.
I mean, a therapeutic, right, is delivered to the patient because it has a beneficial effect on experience. But does that beneficial effect come from the metabolite, or does it come from the drug itself?
So I'm aware of some case examples, but I don't know if we know it systematically.
So there's a lot to be learned from this.
Yeah.
Hi. I was wondering regarding the mice in the beginning, you spoke about them being germ-free and then being colonized. Did you use any approach to test for a successful colonization of the mice?
I mean, so we do tests of like the the number of the CFUs, right? The colony forming units and the feces. So we can estimate how much what is the microbial load in the feces. We can also estimate it for the intestine when we perform the final measurements. So basically in this case, I mean, when it's one microbe, if it's monocolonized, it's usually not a problem. If you work with synthetic community, then it could be that some of the species are not colonizing, but usually the synthetic community and the single species we worked with were engrafting well in the germ-free mouse, and we tested it with calculating microbial load in feces.
Okay, and the introduction of the bacteria was oral?
Yeah.
Or- I have one more question, but it's general, sorry. So my question was, what do you hope the biggest contribution of your work would be in the future?
So, I mean, I think what we're striving for is to really understand the mechanisms of microbe-host interactions through metabolites, being drugs or be dietary compounds. And I think a little bit what we discussed now also throughout the talk and discussions that, if we understand the mechanisms, we can try to come up with strategies to affect this process, right? Be beneficial, like either promoting host health by, introducing microbes that are, or supporting microbes that are promoting health-related metabolites or affecting drug therapies. So I think we need to start with microbial metabolism and metabolic interactions with the host before we can use this information to affect human health.
Okay, so we have a final question here. I hope it's good.
After this,
No pressure.
no, it will be very simple. When you divide the intestine in several parts, do you check for the presence of bacteria within those several parts? Because it is known that bacteria can go into the small intestine while the mice are healthy, and then the loads increase until the end of the. Did you check that?
Yeah. So here we didn't check the densities, but we did sequencing to see which microbes were present in each of the section, and we do see differences between small and large intestine. The large intestine, it's quite stable along, like in different sections. So the composition is different between small and large, but within the big section, in the smaller sections, the composition is very stable. But that's a good point. For us, we actually, to simplify, we focus on microbes that are in the large intestine, because microbes that are in the small intestine can be doing something completely different, and that's something also to potentially consider in future studies.
Okay, thank you very much.
Thank you.